All right. Hello, everyone. So today I will talk about neurodegeneration from a circuit point of view. So our central question is not how much the structure is lost, but also the impact of this loss on the population activity. So I will approach it from the spiking neuronal network point of view, because it would give us a controllable framework to administer some degenerative rules, and in parallel we can record the activity dynamics. So let's start from the biological motivation. So neurodegenerative diseases are prevalent, and they pose a major challenge on us worldwide, economically, medically, and in various aspects of our lives. So yeah, they are very specific in their region of interest. So they are not randomly attacking any region or any neuron. So at a circuit level, some circuits are resilient and some circuits are drifting from the baseline activity. And the question is why? So that leads us the specific questions of the project. So is there any favorable architecture for the resilience of a given network on attacks? And also, what are the effect of this structural change on the activity dynamics? We can estimate them by firing rate, synchrony, and regularity measures. And other is, do we have like a structural descriptors that we could infer the degenerating activity dynamics from? And if this is significant, can we use that for prediction problem? Can we infer activity from structure? So to answer these questions, we need a controllable setting. So the next slide shows the main ingredients. So the first ingredient that we want is healthy baseline activity. So for a microcircuit, we need weakly active, asynchronous, irregular kind of state. The second one is a neuron model. We use simple link integrated fire neuron model. And we need to have, of course, the network architecture where we start using empirical microcircuit. And we expand that to synthetic counterparts by preserving some global parameters of the empirical connectivity. And of course, we need the degenerative strategies. For the degenerative strategies, we have two broad groups. One is based on synaptic removal, and the other is based on neuronal loss. And we tried to include some biologically observed phenomenon like dendritic degeneration or random pruning, or that prunes hubs like we saw in the morning session, even though it's in a higher scale. We include also like two counterparts, like if we degenerate the neurons or synapses, targeting first the hubs, and then we'll have like another version that targets only the leaves or sparsely connected ones, so that we see the effect of these complementary degenerative strategies side by side. So we have 10 degenerative strategies. So we have all we need to start dealing with the microcircuit. So this microcircuit, we've obtained that from Sompolinsky's lab, from one of their earlier work by Landau and colleagues. So they constructed this connectivity from the cell positions and the axodendritic overlap statistics from a rat model, specifically in layer 4 barrel cortex. So as you see, it's not really estimated by a random connectivity, as we see some structure there. Especially the excitatory synapse seems a bit sparser than the inhibitory ones, which are shown in blue. And moreover, we see the degree distribution is heterogeneous to estimate it to random. And we need to have this asynchronous irregular and weak kind of regime to start our degeneration strategies. So implementing those degenerative strategies, the thin ones, the above, this is the result that we have. So the top one shows the synaptic pruning strategies for the 5 variants that I showed you. And the bottom one is for neuronal loss. And the x-axis tells us the loss percentage, or you could take it as like a degeneration stage, starting from the pre-degeneration stage to like the most degenerated version. At each stage, we prune 10% of the elements, be it a synapse or a neuron. So all in all, we see somehow similar kind of pattern, except maybe the hub-based synapses. Obviously, hubs, when hubs are affected, it's clear that we will have a relatively higher effect on the network, as we saw also from Professor Stamm's presentation earlier. Also, it's in a regional connectivity. So that's why we see here mild difference, but in general, that's what we see. So the question now is, if we have some architecture that preserves these global ingredients, and if we have synthetic networks, would we see something like this or similar structure like this? So for that, we need to recruit other architectures. The usual ones are random, small world, and scale-free network topologies. So what we preserve here is the total wiring. The synapses are preserved, meaning the global connection density is the same. And the proportion of inhibition and excitation is again preserved. And also the strength, also the proportion of the strength is preserved. Sorry, I should have mentioned here, we also took this weight, how the weights are taken, usually in the standard setting, for instance, in the works of Brunel, we choose EE and EI to be the same, excitation to inhibition and excitation to be the same. But in here, they used relatively a biased kind of inhibition push somehow. So excitatory neurons excite inhibitory neurons a bit stronger. So we took this weighting scheme throughout until I tell you maybe in one slide that we used Brunel-like configuration. So we preserved that one as well. So then the global scalar for this weight distribution should be a bit tuned so that we'd have almost the same kind of baseline activity with the empirical data. So we started pruning the networks using the neuronal and the synaptic pruning strategies. And this is what we found. So this is again the same description, except that we add, apart from empirical, we have these 3 canonical synthetic networks, and we see a slight drift from the empirical part. So the bluish one is the empirical one, so it's somehow resilient. So why is the empirical slightly resilient compared to the other counterparts? So could it be that there is a hidden biological ingredient within the microcircuit, or would it be that because of this uneven kind of distribution locally? Because as I told you, the excitatory synapses are a bit sparser, but when we took like the global connection density preserved, we didn't also embed this kind of local kind of differences in the canonical networks. Would it be that then? Or would it be some other like inhibition promotion architecture in the empirical network that helped it? So to check that, we adapted these two networks, Erdős-Rényi and scale-free networks, to accommodate a bit stronger inhibition. So we did that in 2 ways. So for Erdős-Rényi part, what we did is like we preserved the connectivity density blockwise. So the connection density for II should be the same as II in here. And the global is, of course, it should be preserved. So that's how we tried to restore some architectures in there, but of course we can't restore the degree distribution as we are in random network. And the way we do that for scale-free I, which is inhibition-promoting scale-free network that we call it, what we did is that we exactly took the scale-free networks that we had earlier, but what we did is like we labeled the hubs to be inhibitory neurons. That's the only difference. So that way we could enhance inhibition. As you see, inhibition is taking higher degrees here, like the hubs are taken by the inhibitory neurons. And as usual, we maintain the baseline activity to be somehow comparable. So the result with this would be something like this. So of course, that preserved or that restored the resilience to the empirical network, and that confirms our hypothesis that empirical network didn't have any hidden biological ingredient there. The main role is like how inhibition is accommodated in the microcircuit itself. And we achieved that with these two architectural kind of designs. So what we saw now is like three networks are behaving the same way, resilient somehow, relatively speaking, and the three others are not resilient. But this non-resilience, is it absolute? The answer is no. We can make this one, the Erdős-Rényi, small world, and scale-free networks resilient if we actually increase the inhibitory strengths a bit higher. Actually, the left one is the one that we used for the first weighting scheme, where there is a bias for excitatory effect on inhibition stronger than the EI strength. But this one is like the well, the name is a bit misleading, standard setting, because we are using that almost throughout the work. But this one is just to use a different configuration where there is no bias. Like there is, we removed this bias, like E to I is the same as E to E in like Brunel setting. So we don't have to even promote the inhibitory synapses by pushing it a bit stronger. So if we strengthen the inhibitory synapses, we can have this resilience. So from this, we can conclude that resilience is highly affected by inhibition, and inhibition can be restored architecturally or by having stronger inhibitory synapses. So if inhibition is the major or the common denominator for this change, can we use this structural stuff for prediction problem? For that, we used ESW, which is the effective synaptic weight, which means nothing but the net presynaptic strength at a neuronal level. Or if we add, if we are talking about the mean of or globally, it would be just the same as the sum of the weighted connectivity matrix. So this is a very simple parameter or variable, because it's just adding the connectivity matrix, but it carries two informations. One, the sign tells us where our regime is. If it's positive, we are more in the excitatory dominated regime, or if it's negative, we are in the inhibitory dominated regime. But all in all, we have our baseline activities in the inhibitory dominated regime. So it's negative, no question about that. Then the value tells us how far, or the level of inhibition somehow, how far are we from the balanced state somehow. So that's why we chose ASW to link with the level of inhibition. So both in the standard setting and in the this one is the biased setting for inhibition push, and the bottom one is unbiased setting, meaning E to E is the same as E to I. So the ASW seems to track the activities, firing rate, irregularity, synchrony, and pairwise correlation, and the synaptic inputs. But we see that these are for different networks. But we see that it lacks generalizing. Ensemble-wise, we can see that there is like rough estimate. For some of them, it's still poor for correlation and variability measuring variables. But for rate and current, for instance, it's good, but it lacks generalization. So can we refine maybe effective synaptic weight in a subpopulation label to promote it a bit better? That requires us to look into different structural features. So we have these blocks, like global blocks, which doesn't focus on subpopulation resolved feature. We just, for instance, K means just the degree, the mean degree of the entire network. And shared is just the number of pairwise shared input counts without taking into account the weight. And spectral radius and effective synaptic weight that I showed you is just this trace. And the others are block resolved features. From block resolved features, we have two categories. One is weight unaware, and the other one is weight aware. So the block degree is weight unaware representative, which is just the probability of connection times the size of the population that we are considering. So this is a good, a simple example that simplifies what's being chosen here. The 4 blocks you see is corresponding to EE, EI, IE, and II blocks. So that should be read like that. For block strings, we just included the weight instead of just the degree. Block variability is just the variability existing in the local effective synaptic weight. So G is just local effective synaptic weight here. So the other one is the motifs of order 2, connected motifs of order 2. So for instance, I will take GII to GIE. So this is one motif. If you count all of them, we'll end up with 8 blocks. So that's what we see here, block interaction of order 2. So as we see, the weight unaware features did not track or are not correlating well with the activity statistics. Here in the y-axis, you see several activity statistics. The weight aware ones are better catch the activity statistics. So how about we take, previously we saw ASW as a global axis. Now how about we take these 16 weight aware variables as a multivariate prediction problem. So clearly that helps, and it predicts well, and also it generalizes well like this. So far what we saw is one state at a time, for instance, firing rate and variability within the firing rate or something like that. And in fact, we can also combine all these functional states, and we can put it as like a vector point representing a network, and we can see how that is looking like in a reduced dimensional space. So any point here shows you, first of all, the top row is just exact duplicate of one another. Only difference is the coloring, and the coloring is different because this one colors based on the degeneration stage from pre-degeneration to the last one. And this one is network ensemble coloration. And this is based on the pruning strategies we have 10. And this one is based on the weight. One is the Landau based and the other one is Brunel based. So this is one point corresponds with a network. So that's just the activity statistics. So those candidates that I showed you, firing rate, correlation coefficient, Fano factor, all together. So this is a way of reducing a multidimensional manifold to like an observable kind of two-dimensional observation. So that tells you which networks are close together. So as we see, it's not really a diffused kind of structure here. Even in the stage, we see the yellowish one is almost the sparsest degenerated stage. They are close together, whereas the pre-degeneration stages are also close together. In the network also, this splits the two groups. Like you see, green, orange, and blue are the inhibitory promoting architectures that we saw resilience earlier in the heat map. So as you see, they are really split, one in group, and two also locally, they are split together. So degeneration actually gives our trajectory a more structured kind of pattern. So that concludes my talk. To summarize, so degeneration response is mainly affected by the level of inhibition in the network. So ASW can give us a global access to have some local kind of local inference for a given local, meaning like for a given network. And weight-aware 16-dimensional multivariate model gave us like better prediction and also like better generalization. So the degeneration trajectories also show like a structured kind of degeneration change. So the biological implications possibly may be like inhibition. This might explain like sensitivity of microcircuits when we damage, when there is a damage in inhibitory neurons. And also it might give us an idea about like failed compensation or sprouting mechanisms. Maybe adding links may not be enough, but these links should sustain or should promote like inhibition to have resilience property, or so that it wouldn't drift away from the baseline healthy activity state. And also it might give us an insight about therapeutic approach. If there is some boosting inhibition could be made, probably that would stabilize back the network itself. So as next step, we need to check several parameters. For example, the cell types, detailed neuron models, and even architectures, network architectures. We can consider several ones. And also we have like homogeneous kind of weighting, like E to E is this much, E to I is this much. How about varying the weights even within a given subpopulation itself? So there are a lot to test, but yeah, that's the next step. So with this, I would like to thank the collaborators, and Arvind, Demian, and Ad are closely supervising this work and working with it. And I would like to thank also you for listening to me. Thanks.